Hello, my name is Krishtavare, and this is my presentation of my research project of the effect of square wave magnetic fields on the growth behaviors of gram-negative Escherichia coli bacteria. So for a little background on this project, I chose to work on this project because for the past few years now, you've probably seen through my blog, I've been working on other projects related to electromagnetism, and this is something that I wanted to use to continue my passion on it. And another reason was this year, at the start of the year, I got severe bronchitis, which ended me up in the ER. And while I was recovering, I had to use antibiotics. And I was thinking that maybe I could create a solution that was, specific to me, that I could implement using electromagnetism, which is why I decided to take on this project. So every research project needs a hypothesis, and my hypothesis was that switching magnetic fields will impact the growth behaviors of gram-negative Escherichia coli K-12 bacteria. So my research objective was to find out whether Escherichia coli K-12 strand can be, affected by the switching magnetic fields. So I had to find out whether it could be stimulated or inhibited, which is the way that I'd be measuring it. And this would be done by manipulating the magnetic field's intensity as well as the time that the bacteria is exposed to the magnetic field. So my project did have some limitations, which I'll list on the screen now, but the main limitations were I didn't really have access to a professional lab or, scientific grade tools, so I had to innovate many of my tools, which I will show later. And another thing was I didn't have access to a very specific tool called an optical density meter, which would have saved me tons of time. And basically what the optical density meter does is it is able to measure the density of the bacteria inside a media, and that would basically show me the growth curve of the bacteria, which is something that I wasn't able to do in this project, but that is something that I will continue in future work and implement into my project. One of the main strengths of my project was the implementation of a Raspberry Pi, which allowed me to automate a lot of the different processes in my project, such as the incubator, which was 100% automatic. And I'll go over it later in the presentation, but here's a quick schematic of my experimental setup. In order to measure the growth changes in my bacteria, I had to plate them on agar plates, but in order to get the bacteria into a countable amount, I had to run a serial dilution on my inoculum. Here's a quick image and diagram of my Raspberry Pi setup. Here's a quick labeled image of my electromagnetic coil that I use to produce the magnetic fields. So after running my experiments, I realized that there was something really peculiar about the data. I realized that the switching magnetic fields could stimulate and inhibit the bacteria, and it was noted that at different intensities and different periods of exposure, the different switching magnetic fields will have different impacts on the bacteria, and that is visible in both of the graphs here. There was a peak change, and it looked like the two values that were presented on these graphs here were almost inverses of each other. After reviewing other scientific papers, it also became clear that other different types of magnetic fields will have different impacts on the bacteria. One paper showed that there was a clear difference in the impact of rotating magnetic fields on gram-positive and gram-negative bacteria. It was noted that gram-positive bacteria experienced a stimulation to 25%, and gram-negative bacteria experienced inhibition up to 17%. And there was it's visible that there was a clear difference in the growth rates of these two cell groups. And this led to a conclusion that the exposure of the bacteria to the magnetic fields was associated with the impact of the cell wall functions of the bacteria, as gram-negative and gram-positive bacteria have clear, distinct differences in their cell walls, and that is visible in the diagram shown on the right. So the study did conclude that switching magnetic fields do instill inhibitive and stimulative effects on E. coli K-12 strand, and further research of this field could lead to the development of life-saving technologies within the medical field, offering as an alternative and direct solution to bacterial disease. But this will include lots of more research, and I will need to find out the impacts of this switching magnetic fields on gram-positive bacteria, as well as different types of bacteria within these two groups.